Welcome everybody to the Fall 2021 Leukemia Committee meeting. Thank you all for taking a couple hours out of your day to join us and to review what we've been up to for the last 6 months and where we are going in the near future. If you can get the next slide. So before I get started into our agenda, I'd like to thank all the people that make this possible, make the work of the leukemia program go. And so I'm going to start with my vice chair, Steve Coutre at Stanford University. He is also serving as the leader of our efforts in chronic lymphocytic leukemia, and as you'll see, with incredible help from many others, including Deb Stevens, who will be speaking. We also have Anjali Advani, who is leading our efforts in acute lymphoblastic leukemia. Jerry Radich, who is the head of our translational medicine. We have Megan Othis, who is our lead biostatistician with support by others in the biostatistical core at CRAB. We have our patient advocate, Gail Sperling, who helps us with protocol development, and you'll see we've been pretty active recently. Boy, I'm doing this all off the top of my head, which is always very worrisome that I'm going to forget somebody that should not be forgotten. I will finish by discussing our operations office. Dana Sparks, who has led the operations office for a very long time, back when I was an executive officer. She has been called back into service to fill in after the departure of Sandy Hita, who has just been an incredible aid to all of the work of the Leukemia Committee way before I took over from Fred in 2012. And we're going to sorely miss her, but appreciate all the help from the operations office as well. And I think those are all the acknowledgements I want to make sure I got to. Let's go through the agenda. Go back one slide. Okay, so we're going to start off with AML, and Laura Michaels is going to lead this. And then we'll move on to correlative studies in MyloMatch and AML-MRD from Jerry Radich. Kendra Sweet is going to bring us up to date on SWOG 1712 and other efforts in CML. The next slide. Then I'm going to turn it over to Kristen O'Dwyer, who is going to lead us through ALL, sorry about that, Kristen, with Anjali Advani, Eli Jabor, Michaela Leitke at Stanford. Then on to Deb Stevens with Drs. Hill and Chadman discussing CLL. And then back to Dr. Michaels for, no, not back to Dr. Michaels. She's shaking her head at me, saying I got this wrong. So we must have moved this to somewhere earlier. Yes. And so when I stop talking, she will put us all straight. And Dr. Michaels, we haven't talked about this, but if you want, I'm not sure who's going to give the overview of MyloMatch, but if you wanted me to, I'm welcome to do that. Or if you're prepared to do it, you can take the lead. You're prepared to do it. Okay. Well, it seems like my work is done until the 5-minute wrap up at the end. And that's my kind of 2-hour Zoom call. So I look forward to hearing from all of my colleagues here on the SWOG Leukemia Committee. And again, thank you all for joining. I will now let the screen be passed to Dr. Michaels.
All right, so I hope everybody can see my screen. I'm going to go to presenter view here.
Can I interrupt you for 1 second?
Please go ahead.
After your presentation, I knew that Gail was on the agenda, but it wasn't listed on the agenda slide, so I forgot to mention her. So after Laura, I would like to come back to Gail Sperling for an update from patient advocacy. Sorry about that. Go ahead now.
No problem. So can everybody hear me and see my slides okay?
Yeah.
Great. So my job is to bring everybody up to date on what has been working and what's being done in MyloMatch and the role that SWOG has taken in that. So we've heard about MyloMatch for several meetings now. I just want to remind you all that the goal of MyloMatch is to put together some cross-cooperative working groups in patients with younger AML, patients with older AML, older patients with AML, younger patients with AML, patients with myelodysplastic syndrome, and investigate the role of targeted therapies in the initial treatment. So this is largely for once patients are going to enter the MyloMatch protocol at their initial diagnosis. But then the goal of the protocol is to provide a way forward for precision-oriented clinical trials that take patients all the way through the duration of their disease. We want to validate the role of flow cytometric-based minimal measurable residual disease. We want to provide an international platform for the validation of new laboratory assays, and Jerry's going to talk about that. And these assays are about assessing both prognosis and response. Our goal is to develop core geriatric assessment and quality of life measurements that inform the interpretation of trial results that are used across all the studies to characterize treatment tolerance and benefit. And then another important part of this national program is to provide mentorship and a launching pad for early and mid-career investigators who are interested in collaborative translational leukemia research. So that's just some of our aims of what MyloMatch is, and there's like layers of work on this, but I wanted to spend the time we have now talking about the trials that are relevant to AML, and which
Laura,
you guys,
can you swap your slides? Sorry, they're not in presenter mode.
let's see here. How's that?
Sorry.
Better?
Yes.
Okay, good.
Sorry to interrupt you.
That's okay. I appreciate that. Thanks, Kendra. So the next step then is to find my slides again here. Sorry. The next step then is to talk about what the trials will be, the ones that are going to be available for everybody to open at their centers. So each patient who enters MyloMatch will undergo a laboratory screening, and this screening will be done to provide rapid molecular diagnostics, and then through an algorithm will then assign patients to the trial that they're eligible for. In the baskets looking at younger patients, those arbitrarily under the age of 60 are called younger here. We're developing clinical trials in the younger basket working group that will have specific trials for patients with core binding factor leukemia, patients with NPM1 or MLL mutated leukemia, patients with intermediate risk leukemia by the ELN criteria, and patients with high risk criteria by ELN. Each patient will go through those, and then laboratory reassessment will again be done and classify patients into either complete remission MRD positive, complete remission MRD negative, or less than complete remission. Those with the complete remission MRD positive will then go on to something called the ERASE study, which I'm going to talk about in a second. Those who are in complete remission but MRD negative will be eligible for appropriate transplant studies, which are being led by the transplant working group that Dr. Stiff and Dr. Perellis are taking on. And finally, those with less than a CR eventually will move to salvage studies. So one of the first studies to be approved by the Leukemia Steering Committee, and which is now percolating its way through towards activation with a hopeful activation date of late 21 or first quarter 22, is our SWOG Sweat study, SWOG-led study that Dr. Schema has told us about, This is patients who are younger than the age of 60 with high risk AML according to the ELN criteria. They would be randomized to one of 4 arms. One is cytarabine daunorubicin given in a classical manner. One is cytarabine daunorubicin with venetoclax given on days 1 through 11. One would be CPX 351. And the last would be a quote unquote less intensive or moderate intensity therapy with aza-ven given every 28 days for 2 cycles. One of the things that'll be clear in these studies is that we will have somewhat uniform supportive care recommendations, and we will be following everyone for MRD, which is the key endpoint here, CR and MRD positivity. The next study, which has been approved by the Leukemia Steering Committee.
Can I interrupt you, Laura?
Please,
Do you mind if I interrupt along the way?
I don't mind in the least. Okay.
1 second to the last slide, just a couple of things. Paul has been instrumental in getting this study off the ground, but he and I have asked Tara Lynn, who is at the University of Kansas, to serve as co-chair, and she's going to be taking the lead. I'm really happy to welcome her to the Leukemia Committee as an active member, and and she'll be moving with this protocol and is feverishly writing the protocol now. As long as I got the floor, I thought I would just mention one other thing, and that is our study numbers. You'll notice we have a kind of cool study number that doesn't look like SWOG S something 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 anymore. So what you're looking at here is what we decided on for the nomenclature of all MyloMatch studies. So MM does not stand for multiple myeloma. It is MyloMatch. It is a tier 1 study, the 1. It's in the younger adult basket, YA, and it's going to be led by SWOG as opposed to other studies that you'll hear about led by other groups. And it's our first trial. Thank you, Laura.
No, that's great. And welcome, Tara. So this study is for the next study I want to talk about is for younger patients with ELN intermediate risk criteria. This is led by the Canadian Trials Group and Dr. Asseline, who remember was one of our trials, one of our co-chairs on 1612 as well. This will be for patients who don't meet criteria for high risk or favorable risk, again, between 18 and 59. There'll be 3 arms here, cytarabine and daunorubicin, cytarabine, daunorubicin, and venetoclax, and azacitidine with venetoclax, again, with complete remission with MRD assessment as an endpoint. Finally, we have, this is called the ERASE study. This is for patients who have already gone through induction, who are found to be MRD positive after induction. And this is an ECOG-led study by Dr. Atala. And this will take patients and randomize them for HiDAC ×2, venetoclax HiDAC ×2, venetoclax CPX ×2, or venetoclax HMA ×2, and then have them proceed to stem cell transplant at that time. And if I remember correctly, Harry, again, I'm going to invite you to pop in. This is for patients before consolidation. Is that correct?
Oh, beginner's mistake. This is their consolidation. So if they are MRD positive, this is what they'll get for consolidation.
Okay, great. Now we move to the second basket, and this basket is for patients over the age of 60 years. And we have a little bit in addition to the entry criteria, again, which is still in progress. But what we hope here is not only will patients get laboratory screening, but patients will also get a fitness screening with a geriatric assessment with measures of frailty or fitness. Now, initially, this screening will not determine assignment because we don't have sufficient validation of those measurements to determine assignment. But what we hope is by folding this in initially, we can develop those that are predictive of treatment tolerance so that we then have more nuanced ways rather than the Ferrara criteria to move people into baskets of fit, less fit, or frail. So once patients are assigned by the basis of laboratory screening or fitness, we would then have a following baskets of trials. On the fit side, we already have the FLT3 wild type study open. This is Jeff Wee's trial that's run out of the Alliance, looking at uprotozolam, or that agent in combination with 7+3. That has met phase 2 enrollment. They're now looking about moving it to its phase 3. It was a phase 2 to phase 3 study. We are looking for and would be open for ideas for older patients who are FLT3 mutated for a clinical trial to go in that basket of older patients with FLT3 mutated AML who are fit, whether or not we have a trial that would be specific to them. We also, I'll talk a second about this arrow indicates one of the SWOG-led studies. This is for P53 mutated AML in patients over the age of 60, and this would be fitness independent. In the less fit or the frail population, we're developing trials for patients with IDH2 mutated AML, IDH1 mutated AML, patients with mutations in MLL or NPM1, FLT3 mutated AML, AML that has no targetable lesion, and AML that arises in patients who've already been exposed to hypomethylating agents. Each patient would then go through a similar laboratory reassessment at the end of the trial period and move on to either salvage therapy or secondary targets. So in one of the SWOG-led studies in this is again furthest along. This is being led by Eric Husselton, who's being co-chaired with Uma Barate. This study is, the endpoint of this study would be again CR plus CRI, that's MRD negative with a target accrual of somewhere between 44 and 97 patients. This study will randomize patients with mutated IDH2 unfit according to the Ferrara criteria to either venetoclax azacitidine or venetoclax plus oral decitabine plus enasidenib. There would be an MRD assessment after 2 cycles of induction, and then a maintenance without randomization, and then additional rates of additional secondary endpoints include the rates of CR/CRI, disease-free survival, and overall survival. And treatment would be maintained until disease progression or intolerability with relatively frequent molecular MRD on bone marrow and blood being done on patients. Now, this study is we hope to duplicate this study in patients with mutated IDH1. As I mentioned, we are awaiting drug availability, and then this study, which has been vetted by the Senior Scientific Council, will go through a final approval and move to the Leukemia Steering Committee. We also have a study that's being led by ECOG with Jessica Altman and Sasha Pearl. This study also has a primary endpoint of MRD negative CR/CRI. And this will, at the current time, takes older FLT3 positive patients and randomizes them to either HMA venetoclax or HMA ven and gilt, with 28 days of gilteritinib and 14 days of venetoclax is currently anticipated. We actually have a planning meeting on this study later on this afternoon. So this particular design is not hammered in stone. And this still, we await drug availability. It has been reviewed by Senior Scientific Council, but would need a second review before proceeding to the Leukemia Steering Committee. We have an alliance-led study with Annie Im and Eunice Wang, who are taking the co-chair lead. And this would be for newly diagnosed NPM1 mutated or MLL rearranged patients, again with a similar endpoint of MRD negative CR/CRI and an accrual of the low 90s or so. In this study, we would have newly diagnosed NPM1 mutated or KMT2A mutate rearranged AML, and they would be, the NPM1 mutated would be randomized. It says here that it's KO539, but that drug is not assured. We're not sure which of the menin inhibitors would be part of this study. So that's still to be determined. That would be a triplet of a menin inhibitor plus veneza versus placebo plus veneza. And then we're also looking at a single arm study for KMT2A rearranged patients with the triplet. There would be bone marrow biopsies with a primary endpoint, again, of MRD negative CR by cycle 2. And then we would continue PRs up to 4 cycles, or people would come off study by cycle 4. This also is going through final vetting, and we wait for drug availability, and then it can percolate up to Leukemia Steering Committee, but it's already been it's already had good review by the Senior Scientific Council. And the last study that's coming through is led by Rory Shallis with co-chairs of David Salman and Meir Zaidan. This is a SWOG-led study. Rory might be on a little bit later, I think, to talk about this in more detail, so I'll just briefly go through it. But this P53 study will have 4 arms. One will be oral decitabine plus venetoclax. One will be oral decitabine plus venetoclax plus a CD47 agent. And we again here have not hammered out which anti-CD4 agent will be part of the CRADA. So this TTI 621 or APRs, those are still tentative. We have oral decitabine, venetoclax plus one of the APR agents, or oral decitabine, anti-47, and one of them without the venetoclax. So again, a 4-arm fitness independent study. This is not one that would just be given to frail patients. It would be given on the basis of age and then response assessments. And this study has a primary endpoint of overall survival. So the status of the younger patient studies, we in the orange indicates studies that have already been approved by the Senior Scientific Council, which are already on the clock to be written, and which will be the first 3 studies of MyloMatch to open. Those are the intermediate risk younger patient study, the high risk younger patient study, and the study for patients who are MRD positive. We have discussed in advanced studies of transplant there's still a lot to be done on salvage, and we are not yet, we don't yet have studies available for core binding factor or NPM1 MLL rearranged. There is, as you know, already a precog study for younger patients who are FLT3 positive with a randomization of midostaurin or gilteritinib, the Selena Lugar study. The status of our older basket, we have 5 studies that have been, concepts have been written. 4 of those concepts have been finalized and submitted. We await drug availability, at which point they will be finalized by Senior Scientific Council and move to the Leukemia Steering Committee. We see this as the second wave, the second wave of studies that will open in MyloMatch. So what's my ask of the committee? If you're interested in working with MyloMatch, please let Dr. Urba know. We still have lots of opportunities. We'd also ask that when the studies are available, that people open them and are help us work out the kinks in the processes. This is a entirely new paradigm for how to treat AML in this country. And we hope it's one that's going to mean a lot more trials for patients and a lot more involvement of a huge diversity of patients in trials, patients who may not get the access right now. I'm of course available to happy to answer any questions. I don't know if I've gone over my time. I will stop my share and take any questions that might have come up in the chat.
Hi, this is Pat. This is Pat Stiff, obviously from the transplant side, and thank you for mentioning there is a transplant group, and we're in the process of starting trials. There's been an alliance trial kicking around for several years of venetoclax maintenance for everybody We're going to target, again, a MyloMatch proposal. Matt Wiedewalt is from Oklahoma, is going to be the study PI. We'll have a SWOG and ECOG champions as well. We've already working forward to this. This is going to be a MRD positive after induction / consolidation, in which venetoclax will be given with a reduced intensity preparative regimen, primarily for older patients, and then maintenance up to day 100 with a day 100 clinical and MRD endpoints. So we hope to have more information for you by the next meeting in the springtime.
Thank you so much, Pat. I also see in the chat here that Tara had some slides to present. So Tara, I didn't mean to railroad over you in the least. I didn't know that, but if you'd like to give us an update of where things stand with the younger high risk patient population, I'd love to see it.
Okay, I will try to go through these quickly. I know we have a really big agenda today, right? And the most challenging part will be getting my slides to come up in the right the right format. And can you guys see these okay?
Yep.
Right format?
Yep, now it is.
The battle. So Paul, Schema, and I have been working on this younger high-risk AML patients, and Laura really gave a really nice overview of where this fits into the whole MyloMatch program. You know, like she mentioned, you know, we're really looking at this idea of looking at patients by their MRD response after the different kinds of induction regimens. And we know that many patients, right, with higher risk younger AML should proceed on to transplant. That would typically be the recommendation, and that patients who are MRD positive pre-transplant will often have worse outcomes. And so the goal of randomizing people to these different induction strategies is to really try to increase that rate of MRD negative complete remission. And so like we just talked about, you know, the patients have to go on to that master screening protocol. And then if you have high risk disease per ELN 2017, you would be assigned to this arm. And these are the 4 arms, and I will just go into a little bit more detail about each of those and why they were chosen. So there will be stratification based on younger patients, 18 to 39, compared to those who are 40 to 59, as well as stratification based on performance status. The entry criteria are pretty straightforward in that you have to, you know, meet the high risk criteria, but not because of FLT3 or a 922. We're trying to make this protocol very user friendly, so we're allowing prior hydroxyurea, empiric ATRA until you know that the patient doesn't have APL. And then, you know, again, right, this is for patients under age 60. We do allow up to performance status of 3, especially if it's disease related, and then an adequate ejection fraction. So the first study arm, arm 1, is really the control arm, a pretty typical 7 and 3, followed by 5 and 2 reinduction if needed. In the experimental arms, the first one that we'll look at is 7 and 3 plus venetoclax. And this is based off of very early phase 1 data by Dr. Stone and his group that looked at younger patients with a non-favorable risk AML. Out of the 10 patients that they've reported so far, they all had a complete remission, and 6 of 8 that were assessed were MRD negative. And so the dosing, we have modified it slightly from the way that he's running his trial, would be with venetoclax on days 1 through 11, and then daunorubicin at 60 milligrams per meter squared and Ara-C at 100 milligrams per meter squared over their standard days. And then the reinduction would be a shorter course of venetoclax plus a 5 and 2 type regimen. Arm 3, the azacitidine venetoclax arm, I think everybody's really familiar with the phase 3 data and the FDA approval of this regimen for patients who are unfit, but using, you know, that they had a CR/CRI rate of 66%. So again, this would be 7 days of azacitidine, and the protocol would have some flexibility if you wanted to do, you know, have the Saturday and Sunday off in your scheduling. And then with venetoclax ramping up to the typical dose of 400 milligrams per day. Arm 4 would be CPX 351. We all know this is FDA approved since 2017 for patients who are older, for patients with secondary or high-risk disease. And looking at that it had a CR rate of, almost 48% in patients who were older and with secondary AML in their phase 3 study. Patients who are in remission would go on to the next appropriate MyloMatch study as those are being written and would come online over time. If there is not another study available, the protocol here would specify some consolidation, and then certainly the option to come off for transplant would be there at any time, and certainly to go on to a transplant study if available. I think it's important to just think about the statistical plan and how large these studies are going to be. So here for the high-risk arm, we would be targeting 240 patients. And, our team at SWOG has been the lead on this study. And so we're using our recent study, 0106, that had a CR rate of 47% in these younger high-risk patients and an MRD negative CR rate of 35%. And so the study is powered to detect a 20% absolute improvement in the MRD negative CR rate. So our target there would be to see that rate increase to 55% in one of the experimental arms. We're targeting 10 patients a month, and then there is a pre-planned interim analysis after 30 patients on each arm have been accrued. And I will stop sharing and see if anybody has questions about that protocol in particular, or I guess anything else from Laura's section.
If I could just step back, Laura, that was great. Tara, thank you very much. Paul and you are leading the way in MyloMatch, and we've been talking about this concept for many years, actually, this initiative, and this has been an exciting year, and I think next year will be even more exciting because these studies are going to actually launch. And it's been an incredible effort by the working groups across the board, intergroup collaborations. My hat's off to Laura for really leading with Jeff Wee, the older AML basket, but really quite proud of the work that's been done here. As you can see, a number of initiatives. We're working closely with the investigational drug branch to bring in industry partners with targeted therapies, except for the study that you noted, the venetoclax-based studies, the CPX 351 younger AML studies. We're still working on a lot of contracting and CRADAs through the NCI. All the drugs will come through the NCI. The NCI will hold the IND for the study for all of these agents, so it makes it easier on each of the cooperative groups. Jerry Radich with colleagues have been forging ahead on the MyloMatch initiative, the MSRP, not the manufacturer's suggested retail price, but the master screening and reassessment protocol, and he's probably going to mention something about that in his discussion. But all of this is coming together, and we won't have a full panoply of protocols when we start, but we got to start somewhere, and we really do think we'll be starting in early 2022. In terms of, there is a question here about MRD assays, and I'm gonna let Jerry address that when we turn it over to him, which I think will be after Gail's talk, but very exciting time seeing all of this come together. The work that's been going on has been incredible in terms of negotiations with industry partners and in the working groups at the Senior Science Council level. I think, as Laura said, what we're going to see here is a new way. What we're building here is a new way of doing clinical investigation in the myeloid neoplasms across the NCTN with Canadian and US collaboration at the highest level. So thanks for all of you for that effort and making this dream, really a dream, come true. We're so much closer this year than we ever have been. Okay, if there are no other questions, the question about how we're going to assess MRD, I'm going to leave to Jerry to address. But if I can have Gail Sperling Sperling share her screen and give us an update on from the patient advocacy.
Oh my God, my dog just decided to act up. I apologize there. Okay, so thank you. Thanks for the opportunity to speak for a few minutes today. I definitely want to begin with acknowledging the tremendous, excuse me, and exciting work that happens in this committee. I think what Harry just said about AML, I have personally found, based on my experience with the Leukemia Lymphoma Society, to be so very exciting. I'm grateful to be a part of it. And I really want to thank everyone on the committee for your dedication and commitment to patients and their families. It means, excuse me. Next slide. So I did just want to provide a really brief update about the SWOG's patient and community advocates. As most of you probably know, this SWOG Patient Advocate Committee launched this program in 2019. Since that time, this initiative has really taken off. It's brought on, we've gained a few advocates. We really now have a group of 10 new advocates who bring diversity and what I consider really close connections to a variety of communities. It's definitely still a work in progress, but when fully rolled out, this new program is going to increase the number of patient advocates from 22 to 32, and we're getting there slowly, but we're getting there. Next slide. So none of us are physicians, but all of us are but all of the advocates are experts. We just bring I like to say we bring a different expertise to the table. Patient and community advocates provide the same service. The patient, and I always like to add the family voice. That's really our goal, to provide the patient and the family voice. Research, advocates know trials and we know the questions to ask. Some of us, including myself, are sometimes challenged by the science and statistics and definitely technology, as I was this morning with my slides. So please keep this in, excuse me, please keep this in mind when we ask for clarification on any step in the concept development. I might not always understand the statistics and the science behind it, but I've always found that the researchers are really great about explaining it to me. So thank you for that. We do bring an experienced patient eye to trial eligibility. Community advocates are fresh and frontline. I think they're out there, they're working with their communities and really know what's going on. They offer a different patient take on these and other trials or other issues that might affect whether patients enroll in and stay on any kind of a SWOG trial. So just a reminder that research advocates are embedded in one committee like I am with the Leukemia Committee and with the MyloMatch as well. And community advocates can get called in to work on a project or offer advice on any committee. So we have diverse backgrounds. Mine is that I worked with patients and families for 20 years with the Leukemia Lymphoma Society, and Bruce Wright has a very different background that he'll share with you in just a sec. I like to think we're always learning from you, the researchers and scientists, and I like to believe that our input and involvement enhances trial development. This slide takes a look at the new quote unquote communities and new community advocates that have just been added in the last year. Bruce Wright is our military victims community advocate and a CLL survivor, and so I think he's going to be great in providing just an update of his current activities. Slide 4, please, next slide. Just a really quick update on what's happening with Team Science. The goal of this initiative, and I quote, is to support robust and consistent advocate engagement across all stages of research. So that's just to make sure that the patient voice, the family voice is being heard across all aspects of the trial. Module 6 was just rolled out. We've all completed that. All the advocates have completed that one. The focuses on building diversity into trials from the up and in alignment with the FDA good guidance practices. So our committee has had much discussion around improving diversity in our trials. Next slide. And finally, advocates are charged with ensuring that the patient and family perspectives are represented throughout the trial design and development. I call this the can it be done, should it be done, can it accrue patient slide. That's my sort of affectionate name for it. But the questions we want to always ask and look for answers to is, are there added costs for the patient? Can we broaden the inclusion criteria? Are there any barriers to participation? Is this going to make a difference to patient care in a positive way, in a negative way? And will the outcome be important to patients and their families? As you all probably know, there's a lot of discussion. I remember that someone giggled about this last time, about the informed consent process and using new criteria to make sure that plain language and patient-friendly informed consents are being developed, and sort of breaking down consent forms into what we consider more digestible parts. There's also been some discussion. I find this really welcome, for inclusion of quality of life related patient-centered endpoints to accompany the scientific endpoints in both phase 2 and phase 3 trials. Obviously, a lot of discussion about financial burden and of course, increasing diversity and representativeness. So unless there's any, why don't we go ahead and have Bruce share just a little bit. Bruce Wright is our community advocate for veterans issues. So go ahead, Bruce.
Hi, everybody. Pleased to be with you today. I have CLL. Thank you to Agent Orange and service during Vietnam. I am the Veterans Community Advocate for SWOG. Along my journey, I've helped 86 veterans get their benefits that they've earned. One of the most recent ones is Bertrand Hushberger out of Stanford. His doctor comes from Stanford for his CLL. And in all sincerity, I'm here today because of you. Because without your work, I wouldn't be alive. I was diagnosed in 2009. I had single agent treatment in 2014. Gave me 7 years of normal life. But as you know, the mole came out, so it's whack-a-mole time. And I'm now in the middle of my 6-month protocol for obinutuzumab again. It was very well tolerated the first time. Only this time, at the completion of that, my doctor is seriously considering putting me on venetoclax. So I'm here from SWOG for anyone in this community and in this setting. Should you have any questions, you cannot call me too much, text me too much, or email me too much. I'm here to be part of the team. Thank you.
Thanks, Bruce. Does anyone have any questions?
Well, thank you very much, Gail. And Gail actually also volunteered to be the patient advocate voice on the MyloMatch Senior Council, and really appreciate her effort in that regard too. It takes another hour and a half out of her week every Friday to be there. So thank you very much, Gail. Okay, let's move on. I don't have the agenda in front of me, but I'm pretty sure it was Jerry Radich next on the list to talk about the MyloMatch initiatives and the MSRP and all that kind of stuff. Jerry? Let's see, is Jerry on the call yet? I don't actually see him yet. Yeah, I don't see Jerry yet. Okay, so let's go to Kendra. Kendra Sweet will present on CML, and I will text Jerry.
Okay. Let me find my slides. Sorry, I was paying attention to something else for a second.
You're on mute, by the way.
Sorry. Okay, sorry about that. Okay, can you guys see this? Can you see the slides? And you can hear me? Okay.
Yes.
Now we're cooking with steam. Okay, so this is super brief. Just a really quick, probably 2-slide overview or update on S1712. Randomized phase 2 trial of TKIs plus or minus ruxolitinib in CML. This study is already ongoing. I think most of you probably already know that. So I'll just give you kind of an overview of the schema, where we're at with it now, and then an upcoming amendment that I just actually got an update on probably like 15 minutes ago. So I'll tell you where that's at and then take some questions if there are any. So schema, for anyone that's not familiar, this is, again, a randomized phase 2 trial. Key eligibility criteria. This is for, obviously, for adult patients with confirmed chronic phase CML. As it stands now, eligible patients need to be on a 2nd generation TKI, nilotinib, dasatinib, or bosutinib. That should be changing shortly, which I'll get to in a minute. No history of TKI resistance is allowed. So anyone who has achieved certain milestones and then lost those milestones, or have failed to meet milestones, those patients wouldn't be eligible. But as long as patients have met all of their desired milestones throughout their treatment course, those are patients who are eligible for this study. But then patients must have detectable BCR-ABL transcripts measured by PCR that are somewhere between, well, less than 1%, but still detectable, so greater than 0.0032%. So at least a complete cytogenetic response, which is less than 1% by PCR, but still picked up by PCR. And then they need to have been on treatment for a minimum of 12 months and on their current treatment for at least 6 months. So that's a question I get emailed to me quite frequently. So just try to clarify that a little bit. Essentially what they, I guess it's basically what I just said, but they have to have been on some TKI for at least 12 months, but it doesn't have to be the same TKI for at least 12 months. So if somebody was on imatinib for 9 months and then switched because of intolerance, because of fluid retention or nausea or whatever it may be, as long as the reason for switching was intolerance, and now they're on dasatinib and they've been on dasatinib for 6 months, they're eligible for the study. They've been on treatment for a total of 15 months. They've been on dasatinib for 6 months. But if they've only been on dasatinib for 3 months, even though they've been on for a total of 12 months on treatment, they're not eligible because they need to have been on that dasatinib for at least 6 months. So a total of 12 months on treatment altogether, 6 months on their current treatment. Eligible patients are then randomized 1-to-1 to either single agent TKI or combination therapy with TKI plus ruxolitinib. They're stratified based on their TKI and years on number of years on treatment prior to enrollment. They continue on study treatment for at least 12 months, or I'm sorry, for a total of 12 months and then come off the study treatment. The primary endpoint of this study is rate of MR 4.5 at 12 months. The total enrollment planned is 84 patients. So far, we've enrolled 57. The doses of the allowable doses of TKIs are listed here. The dose of ruxolitinib is 15 milligrams twice daily. I did write in imatinib here, which is a little premature, but I'm just putting it there because that should be allowed sometime in the next month or so. That amendment has been submitted and approved with some revisions. So that's actually just the last bit of information to throw out there. We did submit an amendment to allow imatinib treated patients. That has been approved by the central IRB with some revisions. The revisions were submitted back. And that's actually the update I just got because I was asking about that earlier today. I think the issue actually came, we sent the revisions back September 22nd. And then I haven't heard anything, but it was Sandy that sent the revisions back, and then she no longer is with SWOG. So CIRB responded to Sandy, and she's not here. So Dana just reached out to them, and it turns out they've just been sending emails to people who have no email address. So Dana's on top of it now. We're going to get the information from the CIRB, and hopefully we'll move this forward in the next, hopefully in the next week or so. so we can get this amendment open and increase accrual and hopefully finish out the study soon. So I'm happy to take any questions.
So the imatinib dose is only 400. What about people getting 600?
Well, it's 300 to 400. I didn't.
Yeah.
Yeah, I didn't actually include 600. It's a good question. I guess I don't see very many people on 600. I never even thought to put 600 in there. That's a very good question, but you're right, it's not included in this amendment. Is that something a lot of people would want to include that we would need to change? Do a lot of people have patients on 600 of imatinib?
I got a couple people that are ready to go if they're eligible. They're in this category.
Really?
Yeah.
Okay. Okay.
I think Pat brings up a very good point. I mean, if they're showing resistance and not hitting the deep responses that you want, you're going to try to push the dose before you add a drug or change a drug.
True. Yeah, no, that's true. I just haven't seen very many people tolerating 600 of imatinib, so I don't have anybody on it, but that is just me personally. So, but no, that's a very valid point. So I think that's something that we can incorporate once this gets approved. I certainly don't want to change it now, but once this gets approved, we can certainly make that change and submit it again.
Remember, we're in the Midwest.
Got it. All right, good to know. No, I will absolutely keep that in mind because that is definitely not even something that came to mind. So I appreciate the feedback. Thank you.
Kendra, I want to appreciate publicly the leadership that you've had in this clinical trial. As many of the committee members can remember, each of the cooperative groups proposed large phase 3 studies for this population. And the NCI said, no, why don't you just do randomized phase 2 studies, smaller ones, and we'll kind of pick therapies that may be of benefit and compare these novel therapies. But the study that you have designed, Kendra, based on your phase 1 work and some of the preclinical work at Moffitt and other places, is actually the furthest in accrual than the other cooperative group studies, which are floundering and close to being closed. So thank you very much for this, and you've done this all during COVID, when getting people to come in on a randomized trial to get another drug added that's immunosuppressive, and you're still getting this kind of accrual because that's pretty impressive. And this has always been a very tough population to accrue for studies, right? Many studies have failed because of this whole concept of what is the clinical meaning of a deeper remission. I think things have changed now as we approach, are more likely to consider treatment-free remissions. So maybe that's why people are more eager to think about getting into deeper remissions. But whatever the reason, thank you for your leadership here.
Thank you. I appreciate that, and the support of this full committee too, because I've had a lot of support, and I'm really very grateful for that. So thank you. Hopefully we'll get this to the end soon.
What was that?
Hopefully we'll get this to the finish line very soon.
Yes. So I have not heard back from Jerry yet. And I'm doing this from memory of that agenda. So I believe next on the agenda is Kristen O'Dwyer and the ALL group. Oh, but wait, we have a minute. Before you do that, Uma Barate is on. And Uma Barate will always be a SWOG member in my heart, okay? UAB, we made that SWOG, OHSU, and then she went to that state school in the state south of Michigan that we're not going to talk about. She's been leading an effort in MPNs. Uma, can you say a few words about the protocol that you and Aaron Gerds have been working on?
Sure, and I believe you're referring to the Ohio State, but that's okay.
Oh, you even said the. You even said the. Oh my, you have already drunk the Kool-Aid. Okay.
You reminded me of the. I wasn't aware of it, but yeah. You want me to share a slide or just give a
Yeah,
real?
share a slide.
Okay, I can do that real
It's
quick.
the slide is generic, right?
Yes, it's generic.
Okay, good.
There's nothing else. So give me 1 second to pull it up. Okay, here we go. I guess I can just share screen. Can everybody see this? Can
We
people
can see it.
Okay.
Yep, well done.
Okay, so I wanted to thank Laura again for getting this group together and getting it, up and running. And as many of you may know, SWOG has is really trying to develop partnerships with large companies that have robust pipelines, and the idea is to hopefully, use a variety of drugs to develop trials for patients that have sort of other malignancies that are not, AML or MDS or maybe part of MyloMatch. So as part of that, we were really looking to develop studies for MPNs, and we have an MDS working group. And based off the work from that working group, we are looking to develop a trial for patients with newly diagnosed MPNs that have accelerated phase, blast phase, CMML, or MDS MPN overlaps. Most of these patient populations get excluded from our MDS or AML studies. And this slide, as you can see, is very generic. And the reason for that is some of these discussions are still ongoing with various companies. And so we're not really in a position to actually put drugs on the slide. But the basic concept that we're working with is, as many of you know, some of these patients present de novo, have not really been exposed to a JAK inhibitor like Rux or fedratinib. And then you have a subset of patients that already have one of these diagnoses, have been on some JAK inhibitor, and then have progressed either to an accelerated phase or blast phase. So the idea is to have novel combination therapies available for these patients. And we are thinking of these two cohorts, as I just mentioned. With cohort 1, arm A, we're considering a hypomethylating agent with a TIM3 antibody as a novel combination. And arm B, looking at the role of a hypomethylating agent with an MDM2 inhibitor as a novel agent for patients that do not have a TP53 mutation. And we know that, a fairly significant proportion of these patients actually will not have a TP53 mutation. So we feel like this would be a good option for these patients. And then lastly, for cohort 2, we're looking at a triplet with TIM3 antibody HMA, along with the JAK inhibitor that the patients already have been on for their prior hematological neoplasm. We're hoping, just like the theme of MyloMatch and the studies that have been discussed, to keep this fairly open, to not prescribe the HMA that patients would be able to be on. They can choose between azacitidine, decitabine, hopefully even oral HMA like Dex-C or Inqovi, because these are all standard of care options that these patients would normally be getting locally. Again, as all of you know, these patients don't really respond that well to HMA. So, we wanted to make sure that there was that flexibility, but the ability to put the patients on a trial. And so in terms of study objectives, our primary objective would be really, would really be looking at overall response rate. And this is where we're definitely working hard on the protocol, because there's a number of different sort of disease categories that are in this protocol, and we would have to use response criteria appropriate for that original diagnosis. We're also interested in obviously looking at, overall survival, symptom relief, progression-free survival, transfusion burden. Since this is an immune myeloid type therapy, looking at those type of AEs as well. Aaron Gerds, who may not, I think he was on the call, but is in clinic. And so unfortunately is not here to talk a little bit more, but he is working very closely with the SWOG. I'm not going to be able to say the name of the committee well, but the one that coordinates looking at patient-reported outcomes to develop some robust criteria for us to be able to really follow those in this protocol as well. That's it.
Uma, thank you very much, and for your efforts in this. I completely agree. This is a population that is not typically involved in industry-sponsored trials, and I think we made a great case for developing this trial. It's quite far along in its development with an industry partner, but not quite no ink on the contract yet or page. So we're just being a little bit more circumspect right now. But it's going to be here before you know it. So thank you very much. And what I love about the new NCTN is that we get to continue to work with valuable partners, people that we've grown to know and love to work with, even though they move to, other places that won't be mentioned.
Thank you. And thank you for the support by SWOG throughout. I've grown up with SWOG, and so it's really great to continue to be involved with SWOG with all these studies. So I really appreciate it. Didn't know if anybody had any questions before I stop sharing.
We're good. Okay, thank you.
Sure.
I'm going to, Jerry has joined us, and Jerry is going to present from our translational committee. And Jerry, I'm wondering, would you mind saying just a couple of words at the beginning regarding the major loss that we have all suffered in the area of leukemia research in the last week? I'm putting you on the spot. You don't have to if you don't want to.
No, it's fine. We just had a long testimonial with about this. So Eli Esty passed away suddenly last Friday at home during the day. He just went down, and the medics were unable to revive him. So it's obviously a horrible loss for all of us. So, with there's a cliche that, they broke the mold. So with Eli, they broke the mold, they hammered it with sledgehammers, and they rolled over it with a dumpster. I mean, there's been no one like him, ever. And I think that for me, the quote that best represents the best was a quote from Will Rogers, who said, "It's great to be great, but it's greater to be human." I'll leave it at that.
He was truly both. We'll miss him. My comment was, ASH will never be the same. The oral presentations will never be the same. And we're just going to have to figure out who's going to be first in line at the mic now.
Yeah. And whoever it is, they have to wear a plaid shirt in his honor.
And call themselves just a simple country doctor.
Yeah.
Maybe we should all show up at ASH in a plaid shirt this year.
That's what I was thinking.
Excellent idea. I'll see if I can find a plaid shirt.
That is a great idea.
I've got plenty of them. I'll send you some.
Okay. Jerry, thanks for doing that for us. I know it was difficult. It's quite a loss. I'll turn it over to you now.
Okay, well, better late than never here. So let me share my screen and kind of do a brief update of what's going on with MyloMatch. And yeah. Okay, choose that. Let's see. So as you know, we're going to have a series of rapid assays with MyloMatch when people both enter the trial and follow up during analysis. And so the kind of the menu that we'll be going to, and kind of a black box enterprise called MDNet, will be flow cytometry, cytogenetics, plus or minus FISH, depending on refluxing on what their cytogenetics show, the targeted gene panel with a less than 3-day turnaround time, and a targeted gene panel for MRD assessment, which is done by a spin-off company from the UW called TwinStrand. So I'll review some of the stuff here that we've got so far. This is what's going to happen when someone comes in the door. They'll have one sample go to the local pathologist to make the diagnosis, and then there'll be a series of samples that will be sent to Brentwood, who left us to go to Children's, the biorepository, and then samples will go to the Hutch for cytogenetics by Min Fang. And then depending on what day of the week is, it will go to MOCA for the genetic analysis or us at the Hutch, because our CLIA lab is open over the weekend and theirs isn't. So the samples that we're going to be getting at diagnosis, the required samples will be bone marrow for the biomarker studies, and then optional studies that will go for banking, and then required studies will be bone marrow for flow and other correlative studies. So again, I went over this where and when, but let's go to some of the nifty details of how this stuff is working so far. So the updates I want to give you is kind of the one thing to tell you is the master screen reassessment protocol is pretty much done. I'm collecting my last comments from members of MyloMatch today, so I'll finish that up over the weekend. Thanks for SWOG for all their help in making this happen. I'll talk about the Genexus testing system that we've done on pretty far in the validation, and it's really working quite well. I'll give you some results on the duplex sequencing that we've done using SWOG 1206 paired diagnostics and CR samples. And then just so you know, the parallel work in the foundation of the NIH on looking at methods to do MRD, that's been funded. And so we'll be pretty much developing new technologies in the pipeline that will be, once we validate them, we'll go over to the MyloMatch effort. So this is this Genexus box. The two components, the big thing on the right is the actual sequencer. The thing on the left was what they have in the sales market, that basically you put your cell sample in and you'll get your DNA out. That actually now is a unit. It sounds great, although they've managed to engineer a unit that's actually slower and makes less efficient than an actual tech. So I don't know how you design a machine to be worse, but it actually is. So we'll be sticking to humans for the time being for DNA extractions. This is the panel of 40 DNA genes, 29 fusion genes, and some expression targets. And these are all now been in the midst of validation, almost done with it and ready to go. And actually, as I'll show, it works really quite well. So on your left here in figure 1 is a standard XY plot of 170 samples that were blindly compared between a next generation sequencing and the Genexus rapid turnover. And you can see that's pretty amazing that you get that type of correlation. So that's fantastic. And then the one on the right is just basically showing that discordance at very low values happen, but you can make the argument that a 2% VAF or a 5% VAF at that level is not meaningful clinically. So I think the first pass, this stuff looks like it's kind of too good to be true. So that's about ready to go. And then the next issue is just whether or not duplex sequencing will be better than flow cytometry in predicting outcomes. So this is, again, I've shown this before. Duplex sequencing, what it does is it basically makes libraries off both strands. And then you only make calls on the complementary base pair in the exact same base pair location. So your error rate for most of these library preps, just from polymerases and the like, is about 1 in 1,000, which is why you can never get more sensitivity less than that, because you're kind of interjecting random errors. But if you do both strands and use a complementary, your theoretical sensitivity goes down to about 1 to 10 to the 7th. And then on the right, it shows this is an example of someone who has a rare T315 1I mutation in CML. And you can see if you get down to about 1% or a little bit less, virtually every base pair has a call. If you do duplex sequencing, which is the bottom panel, basically all the noise goes away. You just get signal. We've done this in AML. So on the left, top left is a dilution of 9 different cell lines with different mutations, and just shows that the predicted mutation allele frequency from the dilution is very similar to what you would expect to see. 2 patients, pediatric patients on the right, showing clones that go well below a level of a VAF of 1% that you can detect prior to their relapse. And the bottom panel is basically what that top left panel is, but showing the specific mutation locations and showing that once you get to duplex, you basically eliminate all the noise you see. So question is does this really work, and how does it compare to flow? So this is work we've had funded and that Megan helped us out with. These are 67 paired samples who are in CR by morphology, but we have flow from Brent's lab on them. So what we found at diagnosis is the most common mutation was FLT3. The average VAF was 31%, and 90% of patients had at least 1 variant. The median number of variants is 2. At remission, we could detect mutations with an average VAF of 0.2%. And you can see we found them all the way down to 0.036%. The most common were some of the CHIP ones, as you'd expect. And 68% of the patients had a diagnostic variant found at the remission site. And you can put everything into a 2x2 table. When I was an epidemiologist in grad school, my mentor used to say that basically all life experiences can be put into a 2x2 table, and this is just another one of them. On the left there is whether you're duplex sequencing negative or positive. On the top, flow negative or positive. These are the remission. You can see the concordance is about 70%. So there's 30% discordance, and most of the discordance, 24% of all the samples, are flow negative but duplex positive by the sequencing method. And in the bottom left, I've kind of summarized these. Of the 15 discordant cases with duplex sequencing positive and flow negative, 9 of those patients relapsed. So it kind of bodes poorly. And here's an example of that. This is various ways that you can define, we're kind of learning how to do this, right? We're learning how deep to make the cuts on duplex sequencing, whether you're positive or not, whether you throw out the chips, et cetera. On the bottom there, you see that the flow cytometry has an odds ratio of 2.5 for time of relapse, 2.2 for relapse-free survival, 2.4 for overall survival. So we've got different ways to make the cuts here. The best quote unquote way is the top, where you look at only non-DTA VAFs of greater than 0.1%, or an NPM of 0.1%. That's just going by the guidelines that some past papers in the ELN have looked at. And you can see, if you believe that, the time of relapse-free survival, and overall survival, the odds ratios associated with those are way stronger than with flow cytometry. So I suspect this is probably going to work. We got money to do, May don't have to remind me, but I think about 200 samples from SWOG from another cohort to bust this out for a validation study. So we're starting to get those samples now, and we'll be able to refine this a little bit more, because I suspect that once we do this, that there'll be certain mutations that you can tolerate at some percentage, and there are certain mutations that no matter how low they are going to be awful. And that's just kind of stuff we'll have to learn along the way. And these are just the curves of duplex sequencing positive or negative. And like I said, it looks pretty darn promising for right now. I think I'll leave it at that.
So there's some people that are positive that don't relapse. Do you have long enough follow-up data on those?
Yeah, we have those are old samples. It was probably, I don't know how long when we lost them finally to follow up. Right?
This is Megan. We have up to 5 years. And I think what's really exciting about those curves that Jerry showed is that we had one time point at remission where we did this duplex sequencing. So I think where you see, it's not perfect, right? We're missing some both ways. Some who are positive don't relapse, some who are negative do relapse. But I think I think we're going to get better with figuring out what to do with the data. There's so much data, but what to do with it. But then also in the real world, we wouldn't just take a patient sample once at their first CR, and then never do it again. So I think part of it is that longitudinal sampling that MyloMatch is going to allow us to do, where we're going to improve our accuracy, or we have the potential to improve it even more.
Yeah, but I think really just to underline what Megan said, I think both the performance of the rapid sequencing, thing where R was 0.98, right? I can't find my way home better than that. I mean, it's fantastic. And then the duplex sequencing, it looks kind of better than I'd hoped, frankly. So I think we're really doing well, knock on wood.
So why do you
Terry,
think?
I know that you feel very, what you'd really like to look at is kinetics of MRD clearance. And so to Megan's point, to get to Pat's question, maybe what we're seeing in those patients who are positive at the end of remission, but still long-term survivors, either we're going to postulate some kind of immune effect on residual disease, or just the next cycle of chemotherapy or consolidation or whatever.
Or the third possibility that we've seen in CML and has been described in some leukemias is that you're actually picking up signal in a lymphoid population. That's been shown in myelodysplasia in AML, that some of these markers are in the lymphoid population, and they're not going to relapse.
Okay.
And we'll figure that out from the single cell stuff.
Well, I want to thank Jerry. He's been tireless in his efforts to make this happen. He's been a great partner with me for years in this MyloMatch initiative. And quite frankly, we would not have convinced the NCI that we can do this if it wasn't for the incredible science that we are going to bring to this initiative. And I thank Jerry and others.
Thank you.
For that.
My pleasure. It's been fun.
Okay, let's move on. I think ALL and Kristen is up next.
Okay, I think Anjali has the slides and she's gonna do part of the presentation and I'll do the second half. But we wanted to start with something special for one of our very important team members here. Anjali, do you have the slides?
Yes, just a second.
Perfect.
Sorry about that.
Oh, that's okay. Well, we do have a very special birthday today for Megan. Don't kill me, Megan, but you
birthday.
What's that?
Happy birthday, Megan.
Okay,
And,
Megan. Okay, hold on, everyone.
I just wanted to just make it a fuss, because what she does for all of us is just amazing. And I don't know how she gets it all in, but she does. And having worked in the intergroup on the ALL team, I know that everybody in the cooperative group thinks that we have the best statistician. So,
Oh, and that goes all the way to the NCI.
yeah,
Because that's what I hear as well at the NCI.
Yeah.
So, okay, now we have 85 participants. I want you all to unmute now. All unmute. Giving you a second to do so. You have my permission.
Go for the best.
And now, we will sing. Please join me because I cannot sing. Go ahead. Happy
Birthday to you. Happy birthday to you.
oh,
Happy birthday, dear Megan. Happy birthday to you.
now Megan, there was a lot of love love in that, but just don't misinterpret.
That was amazingly horrible, but the great thing about a Zoom happy birthday sing-along is that Megan can now blow out the candles and none of us have to worry about her spitting on us.
That song
Happy
is really
birthday,
hard
Megan.
to sing.
Well, thank you, everyone. It is, I was kind of hoping I'd be celebrating my birthday in Chicago, but maybe next year. And to go back to Jerry's mention, I met Eli, I think at my first SWOG meeting. He was sitting in the front row and asked some question that had to do with data. And I started working with him. And I think part of the reason I ended up getting, or I knew I was going to work with SWOG, but part of the reason I got so invested in AML and leukemia was through kind of just like the joy of working with him. So it all kind of comes full circle. But so I don't get to work with him anymore, but I still get to work with all of you. And it's just really a pleasure to be part of this group and get to help the science and work with just such a group of really smart, humble people without big egos who it's just a pleasure to kind of be part of the team. So thank you all.
Thank you, Megan. And you know, Megan, it occurs to me that as speaking as a clinician, you're probably one of the few people that would not be shaking in her boots or his boots as he would come to the microphone and ask a question. You'd probably be able to handle it. The rest of us are like, oh my God, what's he gonna ask? But you,
I shared this morning that I actually didn't know he was a big deal for like years. Like I just thought he was like kind of this eccentric person who I was like enjoying writing papers with. And then at some point, I stuck around for Ash for more than just like my presentation. I was like, oh my gosh, he's like a really big deal. Anyway, he took my husband out to a Mariners game because Eli loves sports and I'm like not a baseball person. He took my husband and my husband's like, here's this guy. He's got all these potato chips. He's got like mustard everywhere. And you're telling, anyway, he's, so yes, I didn't even know enough to be scared because I just kind of show up and judge people as I interact with them. And he was fun and whatever.
and that's it, sums it up for Eli. That's the kind of man he was, kind of person he was. We'll miss him. Kristen, thank you for the slide and the opportunity for me to sing. My wife usually doesn't allow me to do that unless the door is closed, and it was.
Well, it's perfect. So Anjali's going to start off and then I'll pick up at the in the midpoint. Okay.
All right. Can everyone see the slides? All right. So Kristen and I, and if Eli is still on, I know he may have had to get off, are just going to present some of the studies. And this is really on behalf of many members of our team that are shown here, and including both Megan and Anna as well, and Sandy and Dana. So I'm going to first briefly talk a little bit about the studies that are currently open. So the first one I'm going to talk about is A041501. This is the trial being led by Dan D'Angelo of the Alliance. This is for young adults with B-cell ALL, so patients up to age 40. And this trial is basically using the C10403 induction. If patients are CD20 positive, they receive rituximab. And then this trial is randomized for patients to either continue to get chemotherapy or to get 2 cycles of inotuzumab and then go on to continue chemotherapy. And so the primary endpoint in this trial is 3-year event-free survival. The trial is stratified by age, CD20 status, as well as the pH-like signature. So this study so far, the accrual is at 226. 48 of those accruals have been from SWOG, and the total goal is 341 patients. So the trial seems like it's moving along quite well. As mentioned, Patients who are CD20 positive do get rituximab. One of the other changes from C10403 is that patients do get Decadron during induction, and there is a cap dose of pegospiriginase to 3750, and there's no reinduction module. So next, I'm going to talk about SWOG 1905. This is a trial that Nick Papadonakis is leading with me. This is a trial for relapsed refractory T-cell ALL or LBL. This is a phase 1/2 trial. So currently we are in the phase 1 part of this study. This trial is with OBI 3424, which is an alkylating drug that is cleaved in the presence of AKR1C3. And so the reason and kind of the rationale behind this trial is that AKR1C3 is expressed at higher levels in T-cell lymphoblasts. And so Cecilia Jung has done some just beautiful work with us in the company, trying to kind of validate AKR1C3. And I think that'll be really an important part of this trial. The phase 1 part of this trial is a standard 3x3 design. Currently, we are in the dose level 2 of the trial. And so far things have been going along pretty well. We do have an amendment that we're working on that Ryan Cassidy has been really helpful with. He had a patient that was on study, and this kind of brought up some really nice points. And so those are shown here. These patients basically get days 1 and 8, and each cycle is 21 days, and then they also are getting CNS prophylaxis with intrathecal methotrexate. The next trial, and I'm not sure, Katie, are you on?
Yes, I'm on.
Do you want to talk about EA9181 since you're on?
Yeah, sure. So
and I can move the slides for you if you'd like.
Perfect. So this is a phase 3 randomized trial comparing the combination of steroids plus TKI with either chemotherapy or blinatumomab in newly diagnosed patients with BCR-ABL positive disease. As you can see on the left of the slide here, in step 1, there's a steroid run-in phase with a TKI and steroids. Patients are between the age of 18 and 75. And then they're randomized to either receive chemotherapy with a backbone of hyper-CVAD, or it's modified hyper-CVAD in patients who are over the age of 70 or considered young and unfit, or to TKI and steroids plus blinatumomab. If patients are MRD positive, then they are reinduced with the alternative regimen. So if they received chemotherapy up front, then they get reinduction with blinatumomab and vice versa. And if they are MRD negative, then they go on to receive either an allogeneic stem cell transplant if eligible, followed by TKI maintenance or consolidation and TKI maintenance alone. And so the trial is accruing. I believe that we're at an accrual of 32, right? Oh, we have some additional slides here. So the primary objective of this study is to compare the overall survival following induction with steroids and TKI plus blinatumomab versus chemotherapy in the combination. The secondary objectives are to compare the rate of MRD negativity at week 15. To evaluate the rate of MRD negativity by treatment arm for those patients who are MRD positive after the first induction and if administered a second induction, to compare event-free survival between the study arms, to assess the toxicities of both regimens, and to describe the outcome of patients who proceed to allogeneic transplant versus those who do not. Like I'd mentioned before, patients have to be between the ages of 18 and 75 years of age. They must be newly diagnosed with BCR-ABL positive BALL. This is confirmed by FISH testing, and there actually will be FISH testing and PCR testing centrally done for the study. They must not have received prior chemotherapy for BALL. And patients who've started imatinib prior to study are allowed to proceed on the study if they've received less than 14 days of imatinib. And patients must not have previously known significant organ dysfunction, as outlined below. And again, as we'd mentioned, overall survival is a primary endpoint. With this study, we anticipate that we'll need about 330 patients. And we assume that the long-term cure rate and overall survival is about 60%, and the median overall survival in non-cure cure patients about 15 months. Like I said, we think that we'll need about 330 patients, and it will take a little over 5 years to complete accrual for this study. Okay, I think that those are the major things. Anything else to add, Anjali?
No, that's great, Katie. Thank you. Did anyone have any questions about that?
Katie, thank you very much. You have been active member of this working group that led to this study getting approved and then activated. A lot of negotiations, a lot of discussion over the years leading to this. It could potentially really change the way we approach pH positive ALL. So I think it's an important study for us, and probably wouldn't have been done by any other. May not have been done by industry, but we're gonna do it.
Thanks so much.
Actually,
Harry.
you know, I'll tell you one thing. I heard from somebody that the data with TKIs and Blina looks so good that we don't need a randomized clinical trial. I disagree. Third-party carriers have very strong opinions on that as well. So thank you for helping us to get the data someday. Thank you.
Thanks so much, Harry.
So next, this next trial I'm going to talk about very briefly. This is actually a pediatric trial that myself and Kristen are the SWOG champions for. This is AALL 1821. This is a trial of blinatumomab in combination with nivolumab for first relapse of B-cell ALL. And this trial is being led by Stacy Cooper. So in this trial, basically, the goal is to really look at trying to optimize treatment in combination with blinatumomab. And there's been some increasing data showing that the mechanism of resistance is due to endogenous T-cell factors, and that checkpoint inhibitors may be able to overcome T-cell mediated resistance to blinatumomab. And so that was the thought behind using nivolumab here. So this trial is going to be limited within our adult population, so it goes up to 31 years of age. This trial actually has, and I'm going to go to this next slide, has a few different arms. I would say for the purposes of our adult group, really for anyone with first relapse, with marrow involvement, those patients are automatically going to go into this group 1, which would be randomizing patients to either blinatumomab versus blinatumomab and nivolumab. The other groups, group 2 and 3, include a chemotherapy arm. And so some of the patients with isolated extramedullary relapse may be eligible, but those arms currently have been on hold. So really the main arm right now is this Blina versus Blina nivolumab. And this trial has been very slow to accrual. So far, the accrual is at 46 patients. And then there is some very nice correlative work in this trial that's being done in the rabbit lab, looking at characterizing the immune response in these patients that respond. So for E1910, this is a trial that was led by Mark Litzow and Michaela Litke. This trial, I did talk with Mark just before this meeting. So this study has been close to accrual, and right now there are not any updates. So still too early really for updates on this trial. SWOG 1318, I'm just going to briefly give an update. So this is the trial that had two arms. One was the pH negative arm with blinatumomab, followed by pump maintenance for elderly patients with pH negative ALL. The other arm was for pH positive ALL, looking at patients, again, that were thought not to be good candidates for chemotherapy with blinatumomab and dasatinib. So for the pH negative arm, we are in the process of trying to get that resubmitted to JCO. For the pH positive arm, we're going to present our data at ASH in a poster. And then we are also working on submitting that as a letter to NEGM. So that's where that is. For a follow-up study for the elderly patients with pH positive ALL, Michaela Litke and I are currently working on a trial with another TKI. So this is kind of in development with one of the companies, which is why I don't have a slide here, because they wanted us to kind of hold off till things were approved. So Michaela and I are working on that concept. It'll be a very similar design to S1318 for the pH positive cohort, but looking at a different TKI. And Jerry Radich is going to do some very nice correlative work for us for that study. And then I think, Kristen, you were going to take it, I think, from here for A041703. Kristen, I think you're on mute, maybe.
Yes.
There you go.
So this is the last of the, I think, the open trials in the cooperative groups. This is a trial led by Matt Wiedewoldt. It's a phase 2 for newly diagnosed older patients to receive inotuzumab followed by blinatumomab for consolidation. And there's a second arm in this protocol for patients who have relapsed disease and they can and who have not previously received immunotherapy. So it's the same treatment, inotuzumab, then followed by blinatumomab consolidation. So those are the two cohorts. There is discussion about adding a third cohort to this trial that would have patients with pH positive CD22 B cell ALL, and this is a planned feasibility study that we've been hearing about in the ALL working group. So we'll have more about that in future SWOG calls once we know that that's a confirmed go forward. Next slide. So this is the schema. Again, untreated older patients age 60 and above that are transplant ineligible receive an induction cycle of inotuzumab. If they go into remission or have adequate cytoreduction, they can receive a second cycle of inotuzumab followed by 4 to 5 cycles of blinatumomab. And cohort 2 is the same with the relapse refractory population, and patients who are in, of course, CR, CRI can go on to allo in this setting. So they completed the 6-patient run and there were no DLTs. Cohort 1 has fully accrued, and cohort 2, the relapse refractory population, has been a little bit slow to accrue, but they at least 11 out of 29 patients. That might be just a little bit more now, but it's getting there. There are correlative studies with this. They are looking at MRD by PCR. They're doing some molecular profiling. And St. Jude's is running some sensitivities to the inotuzumab and other agents of the blast cell population. So this is the proposed cohort 3. It's hopefully going to be either ponatinib and dexamethasone, followed by ponatinib-ino, followed by ponatinib and blinatumomab. So again, a feasibility study for about 15 patients to see if this type of therapy would be worth pursuing in a larger phase 2 trial. Some of the concerns and why this isn't up and running yet, whether dasatinib suppresses T-cell activity in combination with blinatumomab. There has been some publications recently, so and along with ponatinib, but we're clinically, even though in vitro studies have shown that there is some interaction and T-cell dysfunction with the combination of dasatinib and ponatinib, but clinically, anecdotally anyway, there hasn't been, the responses to this therapy by the MD Anderson group has been very good. Next slide. Oh, we can skip that. Keep going. Oh, here we can you go back one slide? There you go. I don't know if Elias is on the call or not, but this is a trial that he designed and will be, he'll be leading with Marlies Luskin from the Dana-Farber. It's a randomized phase 2 comparing inotuzumab plus mini hyper-CVD to age-adjusted hyper-CVAD in older patients with pH-negative B-cell ALL. This concept has been approved by the Alliance, and the protocol's in the last phase of development, and Elias said that this will be open in 2022. Next slide. We'll be quick given the time. So the schema, the eligibility is untreated patients with CD22 positive B-cell ALL, Philadelphia negative, for patients aged 50 years and older with adequate organ function, and patients will be randomized to receive either inotuzumab and mini hyper-CVD or dose-adjusted hyper-CVAD, followed by pump maintenance. Next slide. So just to be clear, patients will, for the dose-adjusted hyper-CVAD arms, if you're over 70 years of age, based on toxicity profiles from MD Anderson, you'll only receive 2 cycles of the chemotherapy and then go on to maintenance pump. And then for patients aged 50 to 69 years of age who are potentially transplant eligible, you can receive up to 8 cycles of therapy, and then, but early discontinuation is permitted to proceed to transplant. So the inotuzumab dose is 0.9 milligrams in cycle 1, and then it's dose reduced for cycles 2 through 4 to reduce the incidence of VOD that was noted in some of the earlier trials done at MD Anderson. Next slide. And this is the dose-adjusted hyper-CVAD regimen that's been used at MD Anderson that is has dose adjustments based on age. Go on. The primary objective is a 2-month event-free survival of the experimental arm as compared to the chemotherapy arm. And an event will be defined as a failure to achieve MRD negative CR/CRP. Relapse from MRD negative CR or death from any cause. Next slide. And again, there will be, what's really important in this study is the management guidelines for age. Patients 70 years of age or older will receive only 2 cycles of chemotherapy, and patients 50 to 69 can receive up to 8 cycles, but you can come off early if either you're going to transplant or there's excess toxicity. You can skip this, I think. This is, yeah, I think there are a couple of these are out of order. Okay, so then S2113 is the upfront T-cell trial that we have been designing with the intergroup. So this trial has been approved by the SWOG steering committee, and it's going to the NCI CTEP Leukemia Steering Committee at the end of November. Next slide, I'll show you the schema, because I know we've talked about this. This is just a Kaplan-Meier curve for the patients who are enrolled on CALGB 10403 to demonstrate that the overall survival by immunophenotype is equivalent between B cell and T cell with a pediatric inspired regimen. Next slide. And the Children's Oncology Group AALL 0434 was their large randomized phase 3 trial that it added nelarabine to their chemotherapy backbone. And the results of this trial were the arm that received the nelarabine plus Capezi style methotrexate versus high dose methotrexate had the highest 4-year EFS. So these two trials have informed the backbone of the current T-cell ALL trial. Next slide, which will be the AALL 0434 backbone regimen, adding in a randomized way venetoclax and navitoclax. And the primary endpoint will be MRD remissions at day 78 following consolidation therapy. Patients who are MRD negative can then go on to receive interim maintenance with Capezi methotrexate and venetoclax and navitoclax, followed by delayed intensification with nelarabine and maintenance with nelarabine. Patients who are MRD positive after consolidation therapy can go off study and potentially enroll on a daratumumab MRD study that's being designed by Shira Diner and at Northwestern, and that will be essentially a trial of daratumumab to see if it can erase the MRD in this patient population. So the way that we designed this study is to confirm that first there will be a confirmation of tolerability phase to determine that the combination of BCL2 inhibitor therapy along with high dose chemotherapy is safe and tolerable in the young adult patient population. And then the phase 2 part of the trial will be to estimate the addition of the venetoclax and low dose navitoclax in our chemotherapy backbone. Next slide. So again, the stage 0 will be the confirmation of tolerability. We estimate about 10 patients that can be accrued in about 6 months. And patients will be followed through the induction, consolidation, and interim maintenance blocks, which those are the blocks that will include the venetoclax and the navitoclax on days 1 through 14. So if fewer than 3 of 10 patients do not experience a dose-limiting toxicity, then randomization will start. And of course, the toxicities of interest will be mostly related to myelosuppression with ven and nav. And next slide, please. I'm going to wrap up quick because there's more people. So the primary endpoint, again, is an MRD negative rate following consolidation therapy. There's 30 patients per arm, 60 patients in total. We can reuse those patients in stage 0, and we expect that the total accrual will take between 2 and 2.5 years. And we have an 80% power and a one-sided alpha of 15% to detect decrease in the MRD positive rate from 40% to about 15%. Next slide. And then our second primary endpoint will be event-free survival. If our accrual meets the accrual targets that we designed, we will continue to enroll an additional 24 patients or 12 patients per arm for a total of 84 patients, and then we'll be able to evaluate a 3-year EFS. And we expect that the full accrual will take up to 4 years following. We expect to require up to 4 years of follow-up to reach the EFS events based on the EFS of 6.5 years from the control arm on C10403. So the design does require a long time. It's 7 years for total completion. Next slide. The translational medicine objectives will include MRD as measured by NGS, comparing it to the MRD as measured by multi-parameter flow. And Brent Wood is doing all the centralized flow for us. Next slide. This was the trial that I talked about, Shira's, that'll assess whether daratumumab can eliminate MRD in patients in T-cell ALL in CR1. Next slide. Anjali, we should skip a lot of some of these.
Yeah, I'd like to save some time for CLL, please.
Yep. So that's we're all finished with that then.
Thank you. I mean, the amount of work that the ALL group has done has just been incredible, and almost all of it in an intergroup collaboration, which just makes us all stronger and more options for all of our patients when we're working together like this. And again, prioritizing these studies for people in our cooperative groups who have not had opportunities previously. So really excited about all that you're working on, and thank you very much. And we are going to finish with CLL. Deb Stevens, you have the floor. Thank you for all you've done for us in this area, and I'll let you wrap it up, and I'll say just 30 seconds at the end.
Okay, great. Well, I hope CLL is last but not least. Let me get my slides on the right setting here. All right, do you see the right view here?
Got it.
Okay, perfect. So I'll try to keep these updates short in the interest of time. First of all, I'd just like to provide an overview of the current cooperative group CLL studies. Most of the time I'll spend on the SWOG study, which is our frontline study, which is open for all ages. It is for a high-risk population who do not yet meet criteria for therapy, so the traditional watch and wait group. The ECOG Akron study was the frontline study for patients less than 70 years of age, but this study is closed to accrual now. And the Alliance study is the frontline study for the patients older than age 70 or older than age 65 with deletion 17p, and the accrual is still ongoing of this study right now. Of course, you guys have probably heard me talk about this study before, but this study is called the EVOLVE study. It is a randomized phase 3 study of early intervention with venetoclax and obinutuzumab versus delayed therapy with venetoclax and obinutuzumab in newly diagnosed asymptomatic high-risk CLL patients. As you all know, we currently take a watch and wait approach to patients who are asymptomatic at diagnosis because prior early intervention studies with traditional chemotherapy agents did not show an overall survival benefit. But now we have some really nice predictive markers that we can use, and a really nice risk score is the CLL IPI score because it combines both clinical and molecular risk factors, as you can see here on the left side, with the highest amount of points and highest risk assigned to deletion 17p or TP53 mutation. It really highly correlates with overall survival and treatment-free survival. And it's a group, a high-risk group is counted as a score of 4 or higher. And this is our group of interest for early intervention. You all know that frontline CLL therapy has changed from chemoimmunotherapy to targeted therapy. And one of the new frontline regimens is venetoclax and obinutuzumab. It's a nice regimen because it is a limited course as compared to our BTK inhibitors, which are longer term and sometimes indefinite therapy, and leads to a 76% MRD undetectable remissions 3 months following this 1 year of therapy. We found that this regimen is tolerable, it's time limited, it's effective, and so it's an optimal regimen for early intervention. And so our question with this SWOG study is, can early intervention with this regimen prolong survival in the high-risk CLL patients? So we have chosen to enroll patients who have had a diagnosis of CLL within the last 12 months. They must not require therapy by IWCLL guidelines. They cannot have any prior CLL-directed therapy. And again, they must have a CLL IPA score of 4 or higher and or complex cytogenetics, which we defined as 3 or more cytogenetic abnormalities, good performance status, and no GI malabsorption as venetoclax and oral therapy. Here I featured the study schema, which again, we just spoke about the inclusion criteria. These patients will be stratified to high risk versus very high risk and randomized 2-to-1 to early therapy with venetoclax and obinutuzumab versus delayed therapy. And again, this is a time-limited course of 12 months. Our primary endpoint is a lofty goal of overall survival, and we need to accrue 247 patients in order to reach this goal. You can see our secondary and translational endpoints listed here. As far as a status update, we did open in December of last year, and currently we have accrued 11 out of our 247 patients, and you can see accrual by site with several SWOG institutions listed here. And, you know, I do think our accrual has taken a little bit of a slower uptick just related to COVID and the concern for, you know, treating these otherwise watch and wait patients during the COVID pandemic. And so just to note that there are a lot of ways that you can, you know, if you want to enroll someone, there's a lot of ways to kind of extend their time until treatment. Just based on some of CTEP's guidance on delays related to COVID-related problems. We did just pass our first amendment. This does allow for 3 days of prednisone at 50 milligrams prior to the first obinutuzumab infusion in those patients who are randomized to treatment. This is in order to prevent infusion-related reactions. It clarifies some discrepancies in obinutuzumab infusion rates, provides some guidelines for if a patient has a COVID-19 infection, and clarifies some of the timelines between screening, registration, randomization, and treatment. And so, you know, we really, I think this is a really important group to study. And so, you know, please keep in mind that this means you do need to check all of those prognostic factors, including a beta-2 microglobulin, immunoglobulin variable heavy chain, and either FISH or genetic testing for TP53. So I'm going to move on to the next because Dr. Hill actually texted me and he had to leave. And so he isn't going to make it today. But he is the SWOG representative for the Alliance study. And I mentioned this before, that this is the frontline study for patients who are older, and which is defined as 70 years of age, or a new amendment has just allowed patients 65 or older if you have deletion 17p. Primary objective of this study is to compare upfront ibrutinib and obinutuzumab with continued ibrutinib maintenance versus upfront ibrutinib, venetoclax, and obinutuzumab, followed by ibrutinib maintenance for patients with MRD positive disease. You can see the secondary endpoints measured here. I'll show the study schema. These patients enrolled are stratified based on deletion 17p status. And again, they either receive a 2-drug regimen of ibrutinib and obinutuzumab versus the 3-drug regimen that includes venetoclax therapy. In the standard arm, the ibrutinib is just continued regardless of MRD status. But in the 3-drug arm, at 15 months, there's an MRD assessment. And if they have an MRD negative CR, they will discontinue the ibrutinib. If they have anything less than that, they continue ibrutinib. Again, eligibility is 70 years or older with untreated CLL requiring treatment. Again, the recent amendment allows patients 65 or older if they have a 17p deletion, reasonable ECOG status, ANC greater than 1,000, platelet greater than 30, creatinine clearance greater than 45, which is sometimes an issue in these older patients, normal liver function tests as well. Current study status here is that there are 355 patients enrolled out of a target of 454 patients. And so this study is actually getting close to accrual as well. And you can see the distribution here listed by the number of patients accrued, including 50 patients that came from SWOG. So I know, Dr. Shadman, I think you're on the line if you want to present the study.
Yeah, thanks, Dr. Stevens. So this is EA 9161, another frontline study for CLL, really targeting for younger patients, but in principle, same treatment regimens, a randomized trial comparing ibrutinib, obinutuzumab, and venetoclax in one arm, and ibrutinib and obinutuzumab in the other arm. We can move to the next slide. We have the study design. So in the triplet arm, the treatment stops at 19 cycles, and in the ibrutinib and obinutuzumab arm, obinutuzumab was given for 6 cycles and ibrutinib was given indefinitely. The other difference between this study and the prior study, the Alliance study, is the fact that this study did not include patients with DEL17P. Next slide, please. So the study enrolled really well and met the target for enrollment of 720 patients back in April. So currently close to accrual, and we're eagerly waiting to see the results at some point. And yeah, so that's all. Back to you, Dr. Stewart.
Yep, and that completes our very rapid run through of the CLL studies. So, so please, if you have any studies or any patients that you're newly diagnosed CLL, just keep our SWOG study in the back of your mind and let me know if you have any questions about it. And then, of course, the Alliance study for the time being is still enrolling as well.
Thanks, Deb, and team there. So like always, you know, I sat here for 2 hours and just looked at the amazing work of this team, and just so very proud to be part of it. And thank you for all the hard work that you do, pretty much for free for the American people and the NCI. So thank you for doing that. I do want to close just one word, and that is I want to thank our Vice Chair, Steve Coutre, who really helped me, actually did most of the work. That's what it means to help me, did most of the work in setting this up. I think Steve has not made it a secret that he actually had COVID infection in early August and has been having a rough time of it. And that's why he can't be here today. So my prayers go out to him for his speedy recovery. But thank you all, and look forward to continuing our work through the rest of the year and into next. And with that, I think we'll close since we're 4 minutes over time.
Great. Thanks, everyone. Thanks,
Thank
Harry.
you, everybody.
Thank you.
Bye.
Thanks, everyone. Bye.
Bye.
Thanks.
Thanks, everyone.